This is a lecture recording of the nervous system. This is in chapter 14 of your textbook, and we're going to talk about the different divisions of the nervous system and also look at the cells of the nervous system, which we call neurons. So in looking at the nervous system, its primary function is to receive information from the outside world. We call that sensory information, and then to respond to that information, which we call the motor response. So we'll talk about a little bit of each of those pathways, the incoming information, which is sensory, and the outgoing information, which we call motor. So there's two divisions of the nervous system. The central nervous system is where processing occurs. So this is the brain and spinal cord. That's the central nervous system, completely encased in bone. The brain is surrounded by the skull and the spinal cord by the vertebrae of the spine. So that delicate tissue is completely encased in bone. And the peripheral nervous system consists of the nerves. So the nerves come off the spinal cord, and we also have nerves that come off the brain. One example of an important nerve that comes off the brain that you're probably familiar with are the two optic nerves that serve the eyes. So we do have nerves that come off the brain as well as the spinal cord. So if they're nerves, we call them the peripheral nervous system. So they're extensions of the nervous system which end in sensory receptors for receiving information that's coming into the nervous system. So again, the two divisions are the peripheral nervous system, which is made up of the spinal nerves, which are shown here in this diagram, and then we have the brain and spinal cord, which is the central nervous system. So we have nerves that come off the brain. Like I said, the optic nerve is an example of that. Those are called cranial nerves. Cranial nerves come off the brain. Spinal nerves come off the spine. So functions of the nervous system, like I said, the sensory division is information that's coming into the central nervous system, coming into the brain and spinal cord, and that information travels along nerves. So it travels through the peripheral nervous system, which is called the PNS, to the central nervous system. So looking back at this diagram, sensory information, for example, something hot on your fingertip, travels in through the nerves to the spinal cord, up the spinal cord to the brain for processing, for you to tell or distinguish that temperature is hot that you felt out here on the fingertip. So sensory is bringing information from the nerves to the brain and spinal cord. Integration is a term we use for when the brain is processing that information and deciding what to do with that information. So then the brain will send out a response. And if it's, again, using the example of something hot at the fingertip, moving the hand away from the source of the heat is the motor response. And that's what the brain would decide to do by integration, by receiving the information and then deciding that a response is necessary. So it's going to stimulate the motor nerves of the muscles of the hand and arm to pull that hand away. So the motor response goes from the central nervous system through the nerves to the muscles, glands, or organs. So it's the outgoing information. So you can simply remember it. If you have your notes in front of you, you can write some simple notes here. And sensory is incoming information. Integration just means we're processing in the brain, and the motor is the response or outgoing information back out to the organs, glands, or muscles. So when you look at the cells that are transmitting this information, those cells are called neurons. And the other cells that support and nourish neurons that we find in nervous tissue, those are called neuroglia. And another way to remember that is they're like nerve glue. It basically is, these cells are special cells that are the supportive cells of neurons. So the neurons are the ones that are transmitting impulses that are important for sensory and motor responses, but it's the neuroglia or neuroglia that support the neurons. So these are called microglia, astrocytes, and oligodendrocytes, and in the peripheral nervous system are Schwann cells. So these four different cells are examples of neuroglia, which don't transmit impulses, but they support and nourish nervous tissue, and they function to protect neurons in one way or another, and we'll talk about those in a little bit. But these 4 cells are also the cells that can divide in number, unlike neurons, which we have once we reach puberty or in adulthood. Our nerve cells don't really reproduce or repair themselves very well. So we can have a little bit of nerve damage and recover, but if entire neurons are destroyed, recovery is impossible for those neurons. So there is some ongoing research that says there is some neuron regeneration, but it's very limited. So that's why people that have had brain damage or strokes or a spinal cord injury, sometimes that damage is permanent because those neurons cannot repair and recover from major losses of either blood flow or just being damaged through trauma. So these cells, these 4 cells, the neuroglia, these can replace themselves, and they are the ones that are responsible for forming brain tumors. So when people have brain tumors, it's not the neurons that are growing out of control, it's these 4 different neuroglia cells that are growing out of control that causes brain tumors. So different functions of the neuroglia, these supportive cells, microglia, they kind of are the janitors of brain tissue. They clean up the bacteria and debris that might be entering into the brain or spinal cord. Astrocytes provide structural support. They hold neurons together. They keep them in place. Oligodendrocytes form protective myelin sheaths around neurons to protect them and help transmit impulses more rapidly. It's kind of like an insulation on the axon of neurons, which we'll talk about in a little bit. And Schwann cells do the same thing that oligodendrocytes do, except they form myelin sheaths around neurons in the peripheral nervous system, which is around our nerves that extend off the brain and spinal cord. So oligodendrocytes we're only going to find wrapping around neurons in the central nervous system, which is the brain and spinal cord. And the Schwann cells we're going to find forming, that insulating covering over neurons in the nerves. So if we look at a typical neuron, then there's different types. There's sensory neurons that are carrying information from the sensory receptor to the brain or spinal cord for processing. And these sensory receptors are really important because they give us important information about our environment, about temperature or pressure, taste, sight, hearing, the special senses, which we're going to talk about in chapter 15. So the 3 types of neurons then, we've already talked about. The sensory neuron brings information from a sensory receptor to the brain and spinal cord. Then the interneuron is found within the spinal cord. It's kind of the connecting neuron from the sensory neuron from the outside reaching up into the brain for processing. And then the motor neuron are the neurons that carry information from the central nervous system back out to the cell that's going to respond. For example, a muscle cell or a gland or an organ. Those are called effectors. Effectors are any cell that is in charge of a response. So, for example, if you pull your hand away from a hot flame, the effector cell in that case would be muscle cells because they're able to respond by pulling the arm away. If you're talking about shining a light into to a person's eye and their pupil constricts, the iris, which is the muscle around the pupil, would be the effector cell because it's what actually responds to the light being shown into the eye. So the effector cell is stimulated by a motor neuron, which is coming from the brain and spinal cord to those cells to respond to some change in the environment. And then if we look at a typical neuron, there's 3 parts to a neuron. There's the cell body, the dendrites, and the axon. So if we look at a structure of a typical neuron, we can see one here. The cell body is the enlarged end of this neuron, and the dendrites are the extensions of the cell body. They're the receiving portion of the neuron. So they kind of act as antenna, receiving information and bringing it into the cell body. So these are called dendrites, and they are the receiving end of this neuron leading to the cell body. And then we have the axon. The axon is the highway carrying that impulse from the cell body to the axon terminals, and there might be a muscle cell here, some effector cell that's going to be stimulated as a result of an impulse traveling to the axon terminal, or it might be another neuron if it's traveling, say, from the brain down the spinal cord and out down the arm, there might be a series of these neurons, connected near one another. The space between one neuron and another neuron we call the synapse. So they're not directly touching, but they're very close together. So the axon terminal might be near the dendrites of another neuron, and that space between them is called a synapse. So one more thing I want to mention is we look at the axon, we'll see that there's this little wrappings, these insulated coverings of the axon, and that is called a myelin sheath. So that insulating covering, which helps speed the conduction, is called myelin or myelin sheath, and that is only found on the axon of neurons. So we have the dendrites out here, the receiving end, the cell body, which is for processing, and then the highway for carrying that impulse to the end of the neuron is called the axon. And the axon leads away from the cell body and has this insulated covering. Not all axons in nervous tissue have a myelinated covering, but many do, especially those neurons that make up your nerves out, down your arms and legs and serving the skin. Those are myelinated for rapid impulse conduction. And then again, we have the axon terminal at the end of the neuron. So the myelin sheath, we said, covers some axons. If it's formed around a nerve in the peripheral nervous system, those are Schwann cells that make up that myelin sheath. And if we're looking at a neuron in the brain and spinal cord, those are called oligodendrocytes, those cells that make the myelin in the brain and spinal cord. And there's certain regions of a axon that is not covered with myelin, and that's what we see here. These little bare spots on the axon, those are called nodes of Ranvier. It's a French word, Ranvier, instead of Ranvier. Some people read it that way, but it's actually Ranvier. So these bare spots are where the impulse can jump from node to node to node for rapid impulse conduction. So that's the nodes of Ranvier, bare spots on the axon for rapid impulse conduction. Almost like if you were crossing a river and you wanted to get across quickly, you would jump from stone to stone to stone, and that would be like the impulse traveling from node to node to node. So if you look in your textbook, you will find a nice picture of a axon that you can label. And I would recommend, drawing a picture of a neuron so you have something kind of to look at as we follow along here. It's always good to do a little bit of artwork. I know when my daughter took anatomy and physiology in high school, she was actively doing that. And it really kind of brings the information home and makes it more personal when you can draw your own picture and label these structures that I'm talking about. And you have some nice pictures in your textbook as well to guide you. So, like I said, the nodes of Ranvier are those spaces between the myelin wrappings on an axon. And when we look at gray matter versus white matter is white because it has myelin coating those neurons. So we know that white matter is formed in the outer portion of our brain, and we see white matter around our nerves, in our nerves, because a nerve is simply a bundle of neurons that are myelinated, and gray matter is unmyelinated. So when you look at cross sections of brain tissue, for example, gray matter is simply unmyelinated portions of neurons, which are cell bodies and dendrites. Those do not have myelin because they need to be exposed so they can be stimulated and process information. So multiple sclerosis, maybe you've heard of, is a disease in which myelin breaks down. The body attacks myelin in the brain and spinal cord. So this is a disease of the central nervous system, not of the nerves, but of the brain and spinal cord. So every body system is affected with people that have multiple sclerosis. It's a disease that comes and goes, but it is progressive, which means that even though it comes and goes, it does get progressively worse over time. Some people can live a very long time with multiple sclerosis if they take their medications and they have a mild form of the disease, and other people decline very rapidly. It just depends on their individual response to the disease and what age they acquire multiple sclerosis. The younger someone is when they acquire multiple sclerosis, it seems the more mild the disease, and the older they are when they acquire it, it seems the more aggressive the disease. So it's really a complex disease, and there's a lot of ongoing research as far as the causes, because it's really it's an autoimmune, again, disease where we don't quite know the cause, but there's certain things that some people have in common, but then again there's a lot of variability as well. So it's a difficult disease to diagnose, and there's some really good effective treatments out there, and it's important that people do stick to their medication regimen to prevent that attack of their body on myelin. So if we see that we have damage to a neuron, if the myelin sheath is intact, then that nerve can regenerate. So that's important for people that have maybe some nerve damage due to some slipped discs in their back. As long as they do the surgery and repair the damage, to the alignment of the spine to take the pressure off the nerve, sometimes those nerves can regenerate. So it's amazing, that the body can heal, and the myelin sheath plays a really important role in helping axons recover from damage. So the stimulus in the nervous system is called an action potential. So an action potential is due to ion movement into and out of neurons, and the stimulus that results from that movement of ions is called an action potential. And the two primary ions we see involved in making stimuli or developing an impulse in a neuron is sodium and potassium. So in order for an action potential to occur, though, we have to see enough ion movement to change the voltage inside the cell to negative 55 millivolts. So there has to be enough stimulus to cause ion movement to reach negative 55. So there's an animation out on Blackboard that you can watch that talks about the action potential and how the action potential is generated. So I recommend that you stop the video at this point and watch that animation. So we talked about how neurons end with these axon terminals, and the axon terminals will either lie near a effector cell, like a muscle or a gland or some organ that is going to stimulate, or it might lie next to another neuron. And in that case, the axon terminal might be near the dendrite or cell body of another neuron in order to stimulate it with an action potential. So the space between the axon terminal of one neuron and the dendrite or cell body of another neuron is called the synapse. So the space between those is called the synaptic cleft. And a chemical will leave the axon terminal of one neuron and diffuse across the synaptic cleft to stimulate the dendrite or cell body of the other neuron. So what we find is the neurotransmitter is a chemical that has a specific effect on a neuron, and it's stored in little vesicles, little small round sacs called synaptic vesicles, and those are stored in the axon terminal of a neuron. So what happens then is a specific series of steps to stimulate a neuron, and it's called the synaptic cleft. And there's an animation that I'd like to show you that kind of shows the steps in which stimulation of the postsynaptic neuron, so the neuron on the other side of the synapse, to show how that neuron is stimulated. So we'll stop here and I'll show you that animation.